Welcome to Domain 1, Molecular Biology, Genetics, and Wet Lab Foundations. Before we can analyze genomes, study gene expression, or build bioinformatics pipelines, we first need to understand where biological data comes from.
Every bioinformatics analysis begins inside a living cell. Using molecular biology and biotechnology, we isolate DNA and RNA, prepare sequencing libraries, and generate millions of sequence reads. This is the moment where biological molecules become digital information.
We can think of molecular biology as a data pipeline. The cell acts as a database where DNA is the master storage, RNA is the dynamic query, and proteins are the final execution result. Biotechnology extracts this information, and bioinformatics interprets the data. The central dogma describes the flow of information from DNA to RNA and protein. In practice, biotechnology exploits these pathways, such as through RT-PCR, while bioinformatics uses computational abstraction, like protein structure prediction, to reverse engineer function from sequence data. Genomic architecture involves complex coding elements like exons and introns. Alternative splicing expands the functional proteome. If a splice site is mutated, bioinformatics analysis of RNA-seq data can detect this by identifying anomalous read coverage peaks in intronic regions. Epigenetics acts as the regulatory hardware of the genome. Chromatin remodeling between closed heterochromatin and open euchromatin controls gene access. Technologies like bisulfite sequencing and ATAC-seq allow us to capture these states for computational analysis. The wet lab interface starts with lysis and isolation. The goal is to break the lipid bilayer while protecting nucleic acids from nucleases. Common methods include enzymatic, chemical, or mechanical disruption, depending on the sample type, to prepare nucleic acids for purification. Choosing the right extraction chemistry is vital. Silica columns, magnetic beads, and phenol chloroform each have unique benefits and trade-offs regarding speed, yield, and safety. A failure here, such as chemical carryover, can inhibit downstream enzymes and result in data loss. Short-read sequencing technologies, such as those using bridge amplification, generate billions of reads simultaneously. While they offer high accuracy, they struggle with repetitive regions and structural variants, and they have an error profile often biased towards substitution errors. Long-read technologies like PacBio-Hi-Fi and Oxford Nanopore are game-changers for resolving complex genomic architecture. These platforms can span repetitive regions, although they each have distinct computational challenges regarding error profiles and base calling. This decision matrix summarizes the trade-offs between sequencing platforms. Your choice depends on your specific bioinformatics goals, such as SNP calling, de novo assembly, or real-time clinical analysis, considering factors like read length and primary error types. Quality control is essential. We use UV-Vis spectroscopy to check purity in electropherograms to verify integrity. For example, degraded RNA input can guarantee 3' bias in downstream results, which would artificially skew any differential expression models. Digital quality control, such as FASTQ uses Phred scores to evaluate base call accuracy. Because sequence quality often degrades at the 3' end, computational trimming is required to remove this noise before performing read alignment. In the lab, every decision counts. Whether it is cleaning a sample to remove salt carryover or selecting a specific sequencing platform for complex genomes, the rule remains. Bioinformatics cannot rescue terminally flawed chemistry. Bioinformatics synthesis involves more than just programming. It requires statistical rigor to manage large-scale data and the application of deep learning models for tasks like denoising. Mastery of these fields is built upon foundational texts and constant practice. To summarize, bioinformatics is the digital realization of physical biology. Truly successful analysis requires an uncompromising understanding of the entire continuum, from wet lab fidelity to sequencing physics. Thank you for watching.